science that's happening in the public health space. We've settled on a number of working groups, and it is really this engine room which carries out a lot of the activity that stitches together the work that we are doing within Phage. And I would just remind you again, if you look at the left of my slide, there are certainly a couple of objectives which we try to achieve, and some of them are more measurable than others on the short term. But really, what we're really wanting to see is that public health laboratories exploit and utilize routine sequencing as a way of accelerating the production of health data that can inform a public health response. We have, with each of our working groups, we are very keen to make sure that we promote standards, the ability to reproduce workflows. And I think really in this past year, we've really seen a need to streamline our activities in order to strengthen surveillance, but also improve our capacity to produce a timely outbreak response. And so what I do hope that comes out of the sessions that you would listen to this afternoon would be just again an opportunity for you to see either how we as individuals or as organizations could plug into the activities of Phage. And we are excited by a number of individuals that have signed up as members of Phage. But even beyond that, we have seen very productive engagements with organizations who feel that the activities which they embark on is closely aligned to what we are trying to achieve in Phage. And so I'll definitely encourage you to contact us, sign up, and let us explore ways in which we can work together. And there's certainly a lot that needs to be done. We are currently very much working with SARS-CoV-2 data, but let's not forget that our public health systems are strained globally, far beyond the work that we are doing on SARS-CoV-2. And I think the Phage platform provides an opportunity for us to engage on a number of fronts. There are some housekeeping rules that I just want to go through with you. And the one is that I will welcome each presenter, and I just want to remind you that we have a Q&A session. So if you look at the bottom of your Zoom screen, there'll be a button there you can click on for questions. And I encourage you to submit your questions through that. One of the Phage Secretariat members, Ranga, will monitor that. And at the end of each presentation, she will select a few questions and pose that to the speaker. And so we are conscious of time, but we also don't want to stifle the opportunity to have a discussion after each presentation. And so I think that is going to be the exciting part. We look forward to engaging with you. And thank you very much for being a part of this open meeting. At this stage, I think I'm going to invite our first speaker. And Nawal will there we go. Thank you very much. And so it's certainly a pleasure for me to introduce Emma Griffiths. And she has been an active chair of the Working Group on Data Standards. Emma is based at the Simon Fraser University in Canada. And she and a team in this working group have been working tirelessly at responding rapidly to the crisis that we find ourselves in. And she will be giving a presentation on the work that the Phage group has been doing on building a metadata standard for SARS-CoV-2 surveillance. Thank you, Emma.
Great. Thanks for that kind introduction, Alan. I'll just get my screen sharing going. Can everybody see my screen?
Yep, I can see it.
Yep. Great, perfect. Okay, so yeah, thanks for that introduction, Alan. As he mentioned, there's a lot of exciting projects that are going on in the data structures work group right now, and Josephina and Marcelo will tell you a little bit more about that later on in today's meeting. But as Alan mentioned, in the next 15 minutes or so, I'm going to tell you about some of the work that we've been doing developing a metadata standard to help improve data harmonization and integration for SARS-CoV-2 genomic surveillance. So the first thing I'm going to do is tell you a little bit about the challenges associated with contextual data, or what we call metadata. I'm not going to spend too much time on that because I think a lot of people that are tuning in today are probably very well aware of those challenges. Next, I'm going to tell you what's in the data standard and what would be the advantages to you if you considered using it. I'm going to answer some burning questions that you probably didn't even realize that you had, and then we're going to wrap things up. So when we talk about public health genomics data, we're of course talking about the sequence data, but we're also talking about contextual data. And when I say contextual data, I mean the sample metadata, the lab results, the clinical and epidemiological information, as well as the methods information that help us that are critical for interpreting sequence data and for making informed decisions based on analytical results. And it's really this contextual data in combination with sequence data that lets us do things like characterize lineages and clusters, identify variants with clinical significance, and to correlate genomics trends with outcomes and different risk factors. So in any public health emergency, especially a pandemic, getting the right information to the right people is absolutely critical. And that can involve sharing information between groups within an organization, but it can also mean sharing data with trusted partners outside of your organization or with international agencies or with public repositories. And it's the variability in data structure. And when I say data structure, I mean the different fields, terms, and formats used to encode contextual data. It's the variability in that data structure that's found in local or private databases that often finds its way out into public repositories. And that makes contextual data harder to interpret and more difficult to use. So as I just said, the real challenge in different data structures is that it makes information less interoperable and it makes it more difficult to integrate. And that means you have to spend a lot more time cleaning the data before you can start to use it. And that can take hours, it can take days, it can take even longer. And if, say, you're trying to combine information really quickly to resolve an outbreak, you don't have that kind of time. And so best practices for data management, for data stewardship, and for data structures are all really critical parts of SARS-CoV-2 sequencing and analyses. So you may already be aware that there are existing metadata standards already. And so you may be thinking to yourself, why has this group of people gone to such great lengths to create another one? And that would be a very fair question. But it's precisely for that reason, because there are different public repository requirements, there are different minimal information checklists that exist, but none of them are quite fine-tuned for public health or for SARS-CoV-2. And often they're implemented in an ad hoc manner. Now, in Phage, we have a lot of folks that are involved in SARS-CoV-2 sequencing initiatives, be they large scale or on a smaller scale. And very quickly, we realized that we needed something more fit for purpose. And so the Phage data standard has been developed by experts in public health, in bioinformatics, and in data standards development. And increasingly, the standard and the associated materials are being adopted by different databases and platforms all around the world, including Baobab Lens that Dominique will tell you a little bit more about very shortly. Okay, so at this point, you're probably like, that's great, but what's actually in the standard? So the Phage contextual data standard is made up of a number of different standardized fields and terms to describe a lot of different types of information, including different kinds of accession numbers and identifiers, sample collection and processing, host information, so where the host is from, what the host was, different kinds of exposure, reinfection, and vaccination information, sequencing methods, fields to describe bioinformatics processes and quality control metrics, fields for specifying lineages and variant information, pathogen diagnostic testing details like CT values, and fields to capture provenance and attribution so that data providers are properly acknowledged. Now, all of the fields and terms were sourced from different case report forms, international and national, public repository requirements, existing metadata standards, and extensive literature reviews. We've tried to map all of the elements in our data standard to existing metadata standards, and where possible, we've tried to reuse fields and terms that already exist. Because we're not about reinventing the wheel, and interoperability is one of our prime objectives. You can also find the specification at the link in the bottom left-hand corner of the slide. And feel free to contact us if you want to get that information again later. So in Phage, we're not just about developing standards, we're also about developing the tools and the resources to put those standards into practice. And so one of the first tools that we developed was a metadata collection template. And you can sort of see a bit of a snippet of it on the screen here. And you can see there are different fields and they're color coded. So the yellow fields are what we consider to be required. The purple are recommended and the white are optional. And the required fields are prioritized for surveillance. You can also see that a number of fields offer pick lists of standardized vocabulary, and there's also guidance to help structure format for things like dates. Now, you can only see a snippet of the collection template here, but this actually has over 100 fields in it. But the key thing to remember here is that you are in control of your data. You don't have to use the whole thing. You only have to fill in as much of this as you want and as much of it as you need. So it is a spreadsheet. You can just snip out any of the fields that you don't want to use. If you only want to use the required fields, if you only want to use 20 fields, you can custom the template that you use to your needs and ignore the rest. But the key thing to remember here is that everybody that's using the template is picking and choosing the fields that they need from the same pool, and that's what helps to contribute to interoperability. We also have a number of different supporting documentation. So for example, we have a field level reference guide that comes along with the collection template. So you can see at the bottom of the image there, the first tab is the template, the second is the reference guide. The reference guide provides definitions for all of the fields so you know what information we're asking you to put in there. It provides additional guidance for formatting, and we provide examples so that you can actually see what we mean, what we're asking you for. We also have a curation SOP. So this is a set of instructions that tell you how to get started, how to curate your information, and to generally implement the standard. If you are looking to submit data to a public repository like GISAID or the INSDC, and we hope that you will, we've also included step-by-step instructions for how to perform those submissions. These are all found on protocols.io. We're also working on building some instructional videos that will be embedded in those protocols just to make your life easier. Now, you may notice that the submission forms for these different public repositories use different formats and different fields. Don't panic. Our step-by-step instructions tell you how to go from the Phage template to the different submission requirements. And the key thing to remember is that while we have little control over the fields in these submission forms, although it has to be said that we're currently working with NCBI to create a SARS-CoV-2 specific biosample form, that by using the Phage template, we can help to structure and harmonize the information that goes into those different variable fields. And this is a really critical aspect of being able to clean up and harmonize information in the different repositories. Okay, so now some rapid fire quick facts. We've already discussed this a little bit. Do you have to use the entire collection template? No, you do not. You only have to use the parts that you need, and we've highlighted the most important bits for you in yellow. Is this standard just for human and clinical samples? No, it's for all the samples. So if you're doing environmental sampling, if you are looking at different hosts like bats, pangolins, cats, dogs, cows, whatever, we've got you covered. So it's for all the samples. Do you have to share your contextual data? No. As I said, you have all the control in this situation. You don't have to share a single thing if you don't want to, but you can if you want to, and standardizing and making your information more interoperable will help you to do that. What happens if the pick lists that we offer don't have the terms that you need? This is a great question that we get asked quite often. One of the things you can do is you can get in touch with us and we'll provide you with the links to do that. Alan already has. The second option is that the curation SOP tells you step by step how to find a standardized term in existing ontologies. So it'll tell you how to go to the ontology lookup service and find a term for yourself. So that sort of empowers you to find the fields and terms that you need as well. Okay, so what can the Phage data standard do for you? So basically, it's a one-stop shop for consolidating data from different data streams. So, if you have your methods information in one place, you have your epi data in another place, you have your CT values in another spreadsheet. This is a one-stop shop to bring all of that information together. And that's really critical because that helps to future-proof your contextual data. It's not just bringing it together, but it's putting it together in a standardized way where all of the fields and terms are defined. So that will help you, even if you're not sharing any information, that will help you remember what everything means down the road when you go to use that data again for different analyses and for different purposes. And finally, of course, when everybody's using the same standard, it helps to increase interoperability. So that helps to harmonize and integrate information across different labs and different databases. Okay, so what I've told you, it was the short version. If you're interested in learning more, our standard was featured on the MicroBinfi podcast last year. So you can pop over to that website and you can have a listen to our episode if you're doing the dishes or something. You can also read more by downloading our preprint. We put that out last year. Stay tuned because we're going to be putting out an updated manuscript very shortly because a lot of things have changed since last summer when we put out our first preprint. Okay, so I'd like to especially thank everyone on the data structures team and all of the contributors who provided feedback and input into creating the standard. Without them, none of this would be possible. Thanks to the Bill and Melinda Gates Foundation for the funding that allowed us to kickstart the creation of Phage. And very, very special thanks to all of you for tuning in and for listening. And I'd be very happy to take questions. Thanks very much.
Thanks very much, Emma. I'm going to just hand over to Ranga, who will just read out the one question that we have currently.
Okay, good day, everyone. So thank you, Emma, for the great presentation and just the emphasis on how you're making the data actually interoperable and also harmonized across the different labs. We have one question here which says, how do we handle labs that jitter their collection dates? Over to you, Emma.
Great, that's a really great question. So we actually have a field called data abstraction. So you can put the notes, you can put notes in there saying that you've added jitter to your dates, because keeping track of data obfuscation techniques is very critical. So go ahead and add that jitter if that helps you to share data. We've also got different fields that specify whether you're sharing, say, for example, a receive date versus a collection date. But that data abstraction field, I think, is going to probably solve some of those issues.
Emma, while we still wait for more questions to come through, I mean, the one thing that one realizes, there is a specific work culture which we all become accustomed to. So if I have done something in a certain way when I collect my samples, it's very hard for me to change. What is your experience been in trying to get people to shift their best practices? While we have a very good data standard, one is always wondering, how do we move this into the public health implementation setting?
Well, so we've been doing that in a number of different ways. One is getting people more comfortable with using it. So trying to make as many, videos, supporting documents. We're currently working on, in my lab, we're working on a tool called the Data Harmonizer that will, it's basically based on the standard, but so you can enter your information with one click of a button. You can format, it'll automate the transformation into, a GIS-8 submission form, an NCBI submission form, the different submission forms. So it's about making it easier, and less time consuming for people to do their work. So when you don't have to, when you're sharing data with your trusted partners and you're, trying to do your data cleanup, that's very frustrating. So if everybody's using the same standard, that saves time there, saves frustration. If, there's SOPs and there's guidance documents and instructional videos, it really eases the transition. And I think showing different examples of successful implementation really makes this more palatable and more attractive to people, especially when the barriers to implementation are low. So when people are hearing success stories and, it doesn't seem hard to do and it's going to save them time and manpower, then I think that's the kind of thing that will help encourage uptake.
Thanks. I'm going to hand the mic to Ranga. I think we have a few more questions that have come in. Thanks, Ranga.
Thanks a lot, Alan. So the next question here says, data abstraction hinders phylogenetic analysis. How can one know the extent of jittering for each sample?
Right. So just by looking at it, you can't really. And you're very right that jitter does, impact your analyses. So again, we would just encourage people to do that in a kind of consistent way and to make a note of that in their data abstraction field. Now, you don't have to share that information with people, but you've written it down so that you remember. And so when somebody contacts you to say, hey, what's up with your dates, you will have that information on hand. But I don't think right off the bat you can tell when someone's added jitter. Yeah, and that's an important problem. I don't think that's going to get solved by this standard.
Thanks for that. Another one from Pakistan. At GitHub, contextual CSV data to JSON format conversion is mentioned. Any words on application to JSON format?
So yeah, we do provide the standard in JSON format as well. So if you want to be incorporating that into different applications, if you're developing software, that will be available to you as well. There's one available right now, but it's for our older version. So I don't suggest you use that. Just hang on a tick. We're developing one for our updated spec, and that should be out really soon.
And then the last question that we have here, this is from Melanie. Are you planning to add your checklist to the public repositories, such as ENA, for validation at submission time?
So hi, Melanie. I'm glad you were able to tune in. We would love to share our checklist with anybody interested in having it. So I will contact you after this and we can have a chat about that.
Thanks, Emma. I'm just looking around the room again if there's any other questions before we let you go. If not, then I'll just say thank you very much for your presentation. I think we certainly see a number of opportunities, certainly with our participants as well. So looking forward to working alongside you on this further.
Thanks very much.
Well, let me then proceed to the next talk. I'd like to introduce at this stage Dominic Anderson. She is a senior researcher at the South African National Bioinformatics Institute. And Dominic is going to talk to us about how the functionality within Baobab Lens has evolved in the context of metadata standards. Thank you, Dominic.
Hi, everyone. I hope everyone can see my screen.
Yes, we can.
Okay, great. So thank you very much for that introduction, Alan. I really appreciate it. And thank you very much to the organizers for the opportunity to present this talk. So what is metadata? Quite simply put, metadata is data about data. It is the associated information that is required to understand and effectively use data. And it is a key to unlocking a resource. Metadata standards therefore establish a consistent way of structuring and understanding data to ensure proper use and interpretation. A metadata standard increases the utility of the data. It supports interoperability, makes it easier to find the data, provides access to the data, and enhances discovery from the data through its reuse. Metadata management, therefore, is the administration of data. It determines processes of how data is integrated, accessed, shared, linked, analyzed, and maintained. The management applies to physical and virtual data formats, and resources are consumed in this task to maximize the potential of an organization's data. In 2006, Hamby coined the term, "Data is the new oil." Michael Palmer then expanded on Hamby's quote, saying that, like oil, data is valuable, but if unrefined, it can't really be used. So metadata management turns raw data into information. And clearly, inconsistencies in data management leads to inconsistencies downstream and compromises the integrity and the value of that data. So I'm going to talk about laboratory operations specifically. So data management is improved by using a tool such as the Laboratory Information Management System. This increases laboratory potential. So if we think of a sample entering the laboratory, that sample is a form of data. The associated metadata, so data of that data, enters the LIMS. The LIMS effectively draws the roadmap for the sample lifecycle, and this creates a valuable linkage between the sample and its associated data. Therefore, standardizing the data makes the roadmap readable in a universal way, and harmonization is incredibly beneficial to interoperability. The Baobab Lens is an open source LIMS. It was developed using the SOPs of a functional biocollection facility in Cape Town. This biocollection facility is focused on the collection of human biospecimens. And as such, the first metadata standard which was incorporated into the system was the minimum information about biobank data sharing. This included standard minimum metadata related to sample donors, disease ontology, anatomical site terms and descriptors, which could all be linked to that sample. This data would specifically facilitate finding of an appropriate resource and subsequent sharing of that resource. In 2020, SARS-CoV-2 literally went viral, and this public health emergency has resulted in a global combined effort. Baobab Lens had to evolve, and we saw the need to incorporate standardization into the system for infectious disease samples. The Phage consortium established the SARS-CoV-2 Contextual Data standard, and Baobab Lens opted to incorporate this standard. We did this so that appropriately structured and consistent metadata could be captured in relation to SARS-CoV-2 research. This evolution of Baobab Lens aims to promote material and data reuse and sharing through interoperability. The loss of information content, also known as information entropy, generally increases over archival time. And by incorporating these metadata fields in the LIMS, we hope that we increase sample and data longevity. Before we could incorporate the data specifications, we had a number of things to consider. and some items were already on our radar from previous experience, as well as valuable retrospective user input. Fields that were mandatory had to be added, but we had to evaluate the other fields to ensure the suitability for a sample LIMS. Baobab Lens is not an NGS LIMS, and as such does not contain the quality metrics associated with sequencing and bioinformatics. However, we had to ensure some overlap because these downstream processes are in fact linked to the sample management processes. We needed to keep the existing architecture of Baobab in mind. Baobab has process flow and default fields essential to function, which still need to be in place. We needed to remember that sample numbers would be high and that infrastructure capacity is not the same in every single setting. We needed simplicity without compromising the goal of standardization. Once we drafted our plan, we began coding, testing, and in some cases, we had to make modifications. Another consideration that we had to take was making sure that this was applicable in a post-COVID world. So in essence, making it more sustainable and future-proof. The fields incorporated into Baobab Lens consist of configurable content as well as hard-coded options. The configurable content relates to fields which could be applicable to other viral diseases. And once this data is configured in the system, a simple drop-down list is provided in the main sample module. Hard-coded options were those which could be considered more consistent, such as the purpose of sampling, specimen processing, and host information such as gender. To assist with ease of configuration, we included an Excel importer with the 6 content types as separate tabs on the sheet. In this video, I'm demonstrating the import of configurable content types from the Excel sheet. The data in the Excel document includes suggested vocabularies that exist within the template, and this includes lab hosts, host disease, anatomical material, organism, and collection devices. The users can also add to this import sheet, and we have mechanisms in place to prevent any duplication of previously imported data. To add a sample of SARS-CoV-2 and its associated metadata, the user navigates to the virus sample tab. We purposely kept this viral workflow separate in the UI to allow for easy navigation. We have base architecture fields in the default tabs and separated contextual data fields in accordance with the template. For sample lifecycle and data, we included repository accession, sample collection and processing, host information, and host exposure. The majority of fields are a simple drop-down list from configured content or hard-coded content. The mandatory fields are indicated by red squares, and the sample cannot be saved into the system without these fields being supplied. All text fields are barcode scanner compatible, and we included the sequencing tab to create a link for sample metrics and sequencing and bioinformatic metrics using the sequence library ID. Again, we had infrastructure and throughput in mind and created the virus sample Excel importer, which contains all of the fields that exist in the system. The Baobab Lens team did, however, take this a step further and created the standalone viral analysis module. This allows for data related to basic laboratory workflows for COVID research to be entered. All 5 modules can be used together or in a variety of combinations, depending on user need and capacity. In all of the sub-modules, high throughput is taken into consideration by ensuring users can add or remove rows, depending on sample numbers for each experiment. We also captured data and timestamps, kit lot numbers, if they are being used, method linkage, and for the viral load determination, we also included some additional fields from the Phage template, such as the gene name. This submodule also includes reference sample information for QC purposes. Submodules for quantification, qPCR, and sequence library prep filter samples with only the DNA or RNA sample type, so that sample drop-down lists are not a hindrance. While we still have work to do, we really hope that this simplifies an important workflow for current need. So where to next? Baobab Lens needs user input to understand the needs on the ground. We know the system, and while we perform our internal testing very rigorously, we can miss errors. So external testing would really be ideal. We want to add metadata for bacterial samples, but we need to keep in mind that more is added increases the size and complexity of the system for end users. This may mean that we need modularity, which supplements a core. One last consideration that we need to take into account is that we should not just have data for data's sake. And this concept of data minimization is becoming more and more important when legislative principles of data privacy are applying. This means that we need to have a shift towards data by design in our digital world. So we have a number of resources available for users, and like I mentioned, we really value user input and suggestion. We also have an updated demo of the system, and you can contact me if you would like to get access. In addition, we are also scheduling a hands-on training session through the Africa CDC in April, and this will be open for anyone who is interested to attend. So I'd like to finally acknowledge the following teams and funders, and I would like to thank the organizers for providing me the opportunity to present this work. Thank you to the audience for your attention, and I'd like to open the floor to questions.
Thanks, Dominic, for that presentation. As we wait on questions to come on the floor, maybe I could just ask you a practical question. So if I had collected SARS-CoV-2 biospecimens during the first wave in 2020, what is the practical way for me to then import my data to this LIMS? You showed quite a detailed breakdown of the type of features that we have, but I'm just curious, as a researcher within the lab, how would I go about navigating how to add all this information? Do you have some sort of mechanism by which I can import this?
Yes, so good question. Thank you, Alan. So yes, we do have an importer. What we did is we made sure that we designed this importer not just to handle high throughput in terms of incorporating samples in very, very large volumes instead of entering them one by one, but we also created this importer so that people who were already collecting data on Excel spreadsheets could literally just put this data into our template and then just import it in one go. And that way it would actually know where to place all of the items.
Excellent. Well, I'm going to just hand over to Ranga. Ranga, you have a question from the audience.
Thanks, Alan. Thanks, Dominic, as well for the presentation. So Sydney has a question. Can you share the link for the demo? Would love to try this out.
Yes, I can definitely share the link for the demo. I'm just not sure, Ranga, how you would like me to go about doing that. Should I put it in the chat or in the Q&A?
Definitely, you can put it in the chat or Q&A, but we can also share with the people after the presentation, the whole presentation today.
Okay, thank you.
Okay, and then more questions are coming in. Is there an easy way to export from the LIMS to submit into other repositories, e.g. NCBI, ENA? This is Melanie.
Hi, Melanie. Thank you for your question. So Baobab Lens does have an export functionality. We obviously need to incorporate the newest developments into that export functionality, but that is something we're currently working on.
Okay, thanks again. So Sydney has a follow-up question. Is there a data exporter or an API for interfacing with other tools?
There is a data exporter. So as mentioned, we have an exporter that will export information to an Excel spreadsheet, but we do also have a working API within the system.
Excellent.
Okay, thanks, Dominic. Alan, or any of the panelists, if you have any questions.
Thank you. I think that maybe at this stage, let us continue. Thanks so much for that presentation. And as has been requested, we will share the link on the chat so that people can get the direct link to the demo. And then just to, again, remind people that there's also a training session prepared for April. And so we look forward to welcoming you to that as you learn to pick up some of the stuff regarding the LIMS software. We are going to shift gears now, and we are going to proceed to our next talk. And we're certainly very excited to welcome the next two speakers. And the first speaker is Josefina Campos, and she coordinates the National Genomics and Bioinformatics for Pathogen Surveillance platform at the National Reference Lab in Argentina. And she is accompanied today by Marcelo Gallos, who heads up antimicrobial resistance surveillance at the Pan American Health Organization. And we are very excited to listen to the work that they have been doing on looking at the harmonization of antimicrobial resistance data. So Josefina, I hand over to you now. Thank you.
Thanks very much. I will share my screen now. Okay, so we are going to explain how we are going to use the harmonization tool in Latin American surveillance networks. I will handle now the word to Marcelo Gallas from PHA4GE, who is going to explain how we do AMR surveillance through the Relabra network. Marcelo, over to you.
Okay, thank you, Josefina. Thank you, the organizers, to invite me to share with you our activities at the Americas. Yeah, the following, please. Next. Yeah, okay. We are doing surveillance in the region to accomplish, to provide information to the different level, at local level, national level, regional level, and global level. The information that we collect used to be useful for all the levels. At local level, for inpatient care, empiric treatment, antimicrobial procurement at the facility, antimicrobial stewardship program, and also for prevention and control activities at the hospital. At national level, the information that we produce in the region inform decision maker or AMR intervention and guide the national action plan to better implementation and evaluation. Inform technical guidelines, surveillance, clinical management, and treatment. And also inform updates to the essential medicine list in the country and give information about the moment that is needed to do some changes in the essential medicine list at national level. At regional level, the information permit us to evaluate trends in the region and to do alerts in the region to prioritize regional strategies to control and also to do advocacy and resource mobilization for the countries in the region. And at global level, also the information that we collect, GLASS, for example, permit to do global prioritization of activities at global level. Next, please. Okay, what we are doing now, yeah, the Relabra is our network, is the Latin American network of antimicrobial resistance surveillance. Starting in 1996, 20 countries are participating through the National Reference Lab. Relabra is a network of networks. Each country have their own network to produce results at national level. And we have data, aggregated data for selected pathogen drug combination that you have at the right part. We have at this moment 15 different type of microorganisms under surveillance in the region. In between 2000 and 2014, we have 7 150 laboratories that are collecting data, and we are near 3 million data at regional level. And we have now a platform that is pleased. I will show you a couple of slides with the information that you can find. Is open for the public. You can find about American, the surveillance of the Americans. In the last year, we start working in Reliable Plus. That is a different type of surveillance. We are changing the paradigm of surveillance, moving from aggruppated surveillance to patient-level surveillance. And we start with blood culture, and these are the right, you can see the means that you are surveillancing blood culture. And this pathogen include also Candida. Yeah, we have 13 countries participating in the patient-level surveillance. Until the moment, we have 3 countries that already share data with us. And we are working also to include Caribbean at the surveillance, because the 14 countries at the Caribbean don't have a established surveillance system. Next, please. Next, yeah. This is part of the visualization that you have. If you would like to go in depth, you can go to this link in the upper part of the slide, and you can see there the pathogen that we have under surveillance and all the results. And you can play in the platform with years, origin, pathogen, species, countries, etc., etc., etc. And you can see the evolution of the resistance, the trends in the resistance in the Americas. Yeah, it's a big platform. You can enter there and play with the data. For the next one. Next one, yeah. Regarding patient-level surveillance, that is our new initiative. We are doing bacteremia and candidemia. These are the data that we have until now, 76 hospitals belonging to 3 countries and 11,000 isolates approximately in bacteria, and also Candida, 3 countries and 30 hospitals, 340 episodes of candidemia. Yeah, we have the information at patient level in that case. The next one. Yeah, we are working also with new technologies. We have the capacity to do whole genome sequence in 12 from 19 countries in Latin America that you can see in green, and at the Caribbean, we don't have any countries yet that is doing a whole genome sequence. These activities that we are doing in whole genome sequence is a joint activity, is a synergy in between Relabra and PubMed Latin America and the Caribbean. Next one. We did a consultation in October last year, and we are working now in a roadmap to solve the problem that we have regarding whole genome sequence. The current situation at the region is that the application of whole genome sequence is just for a specific situation, not in general for AMR surveillance, and because we don't have money to do that. Management of whole genome sequence data on closed platform with decision makers. Then it means that the countries just share the data that they collect for whole genome sequence with their own authorities at national level. Data sharing is a big problem in the region, like in the rest of the world. Future needs that we identify in our activity in the consultation is that the countries need training, specifically in analysis and interpretation of results by bioinformatics, and also mainly in application of the data for public health activities or measures. Standardized data analysis, standard workflow, reproducible results, regardless the platform that is used. Yeah, we have heterogeneity for this at this moment at the countries, and we are moving to have a standardized way to analyze. Then the activities that you are doing is very interesting for us. And regarding the challenge and opportunities, we have a lot of strengths, weaknesses, opportunities, and also threats. COVID is one of them. But one of the things that you are probably more interested is that the use of new molecular techniques in AMR surveillance is a strength in our countries and the region. And the last one, to give opportunity to Josefina to expose the part of PulseNet. We are working now in like transform data in actions. Yeah, this is what I'm doing now in the region. And one important part for us is the strengths of laboratory surveillance, including new technologies, specifically whole genome sequence and mass spectrometry in the region. And nothing else to add. I would like to say thank you to the Caribbean Network and also Latin American Network. Josefina.
Thanks, Marcelo. So the other network we have in Latin America and the Caribbean is PulseNet. PulseNet was established in 2003 and is one of the 7 regional networks within PulseNet International. And one of the aims is to strengthen national and regional laboratory-based surveillance for foodborne pathogens, for early detection and investigation of outbreaks. And as Marcelo mentioned, we started working with whole genome sequence in 2013 in the Americas, especially in PulseNet, but we still aim to standardize a way to do genomic analysis. So far what we have is a lot of tools going around the different countries and the different national laboratories, reference national laboratories. So far we have many AMR gene detection tools, different output reports of detected AMR genes in a non-standardized or comparable formats. This is quite a challenge for Latin America and the Caribbean. We have a barrier in the comparison of results across tools, in the modularity of tools within the workflows, and of course in how to communicate these results among the national reference laboratories and also between the networks, Relabra and PulseNet, for example, where we have some, for example, regional projects together. So having that in mind is where Data Structure Working Group chaired by Emma Griffiths, who works in develop and promote data standards, started working in one tool that is Harmonization, where data standard for bioinformatic detection AMR genes for genomics data is one of the aims of this tool. The idea is to use the genomic workflow with the different 14 tools that are the most used right now and generate a harmonized and a standardized AMR gene report so we can all the users, the different stage of the users can use and share data. Harmonization provides these standardized field names for reporting AMR detection results, but also provides definition, guidance, and ontology identifier to validate these values, automatically converts the value outputs of 14 AMR gene detection tools to a standardized unified report. And this tool also supports the creation of summary reports across tools and data sets in a variety of formats. Tabular, JSON, interactive HTML that is viewable in a browser. And the most important things, especially for low and middle income countries, that is most of the Latin American and the Caribbean region, is that this specification and harmonization is open source and freely available. This is the GitHub to take a look for this harmonization. How are we using this in Latin America and in the Caribbean? So the idea is to have a pilot between the networks, Relabra and PulseNet Latin America and the Caribbean, to explore the utility for improving genomic-based AMR surveillance communication and data sharing among these national reference laboratories in the different countries and in between both networks. So all participants working with genomics in these networks will test harmonization with the different tools with a local data set, particularly Salmonella typhimurium with resistance and sensible genomes and Shigella sonnei. And these tools will support the creation of summary reports across these tools and data sets in these different formats, so we can all the countries share this data and evaluate what is useful for the networks. Sorry. The idea is to have feedbacks from all the countries to evaluate the tool regarding the installation, the use, the useful information to interpret results. And this is not something something minor, because as Marcelo mentioned, in the networks, in both networks, we have different kind of actors, people that works in the clinical side, people that work in the genomic side, and we all need to communicate in an easy and shareable way to interpret this data and bring it back to the networks to use it, and specifically use it into our national surveillance systems. Oh, sorry, this is not here. So thank you all, specifically to the data structure working group chaired by Emma Griffin, especially thanks to Finlay and Ines and all the Phage consortium, especially all of you for listening, and the Bill and Melinda Gates Foundation for finance this. This is part of the working group that's not possible without the contributions of all of them. I particularly want to thank, I have been enjoying very much being part of this working group and learning with all of these people. Many thanks.
Thank you very much, Josefina, and you've in fact finished a few minutes before time. I'm just looking around the room. If you haven't captured a question, feel free to raise your hand and you could ask it as well. But maybe just as a way of starting, you've made, in the first part of the presentation, there was mention made of data sharing and also use in at least 14 countries. And I then started to think immediately of, do you as a community have the same challenges with sharing data across borders? And I was curious if you could speak to that in the context of your projects around antimicrobial resistance.
Marcelo, if you want to mention, then I can answer through
regarding Relabra, we obviously we have problems to share information in the region because it's sometimes very important information that the countries don't want to share in between them. But in our experience at the region, PHA4GE is like a father in the region to maintain the confidentiality of the information. Then we are exploring now how the countries can share this information through PHA4GE in a regional server, but maintaining the confidentiality of this information. Probably all the countries agree, then probably we will get that in the short time. Yeah? But no, we don't have the system yet for whole genome sequence, but we have the system for the rest of the information. Yeah? Then we hope that nothing happened with the sharing information inside the region. We have to discuss if the information could be open for the rest of the world.
Yes, regarding PulseNet Latin America and the Caribbean, so we have a history of sharing data for foodborne pathogens. We started with PHA4GE when we started working, and we have a regional database that is now moved to Genomics for the countries that are doing genomics for foodborne pathogen surveillance. Usually what we do is share all the data between the countries. So all the consultations, for example, when an outbreak is in one country and we have to ask to another country for information, we share that information, and also for PulseNet international consultation. So where there is an international outbreak, they consult to PulseNet Latin America and the Caribbean, and then we respond as a region. We are all very open to data sharing, but sometimes this has to do with the politics in each country and the decision in each country. So we are very respectful of how each country manage their own data, but we share the data among the network.
I'll just put a plug in for the data sharing working group within Phage, and I think I can certainly see a great opportunity for gleaning sort of information from various countries as we all try and come up with frameworks around how do we best share data, and particularly during a crisis such as an outbreak. So I think there is certainly an opportunity. Maybe just a second question, if I don't see any other questions on the floor. You mentioned, Josefina, this pilot which you have started, and I also saw another slide being presented about the need for certain skills as well. And I'm just curious, if you were to think ahead of this pilot, do you foresee any challenges that could potentially come out of this? Is a skill set in the genomic space something that is going to affect this pilot a lot, or do you foresee a few other problems as you proceed with this pilot?
Well, kind of the idea of doing this pilot is to see what are the problems among the skills on that. So with the data structure working group, particularly Emma and Finn has provided a very useful structure for giving to the countries. And as Marcelo mentioned, even there are 14 countries that are using genomics. This is where we have to link efforts. So some people from the Relabra network works together with PulseNet network. Even we are the same countries and even we are in the same institutions, sometimes are different laboratories. So for example, in Argentina, I'm in the genomics laboratory, but we have the AMR laboratory. So the idea is to join the skills and work together in running harmonization in the genomics data they already have and see how that works. This is also an opportunity for the regions to fulfill that. So to see how we work together and how we can communicate the data between those laboratories, AMR and genomics.
Yeah, and I think it might, and I just started to think about our training working group, and I can see a lot of similarities in scoping out the needs, not only in the countries that we have represented currently, but actually to expand that so that one could conceptualize a training program that best fits public health more generally, and then also tailor-made to the kind of needs which will come out of the pilot which you are planning.
Absolutely, yes, definitely for the training program and also for the pipeline working group. So also to standardize some of the tools we're using.
Excellent. I'm just going to check one more time. I think we have a question. Ranga, do we have another question from the floor?
Yes, there's one question that has come through. For data harmonization, do we need to work on the raw data manually, or the tools will take care of the processing and gives us the harmonized report?
So the idea is to run all the tools and then process and have one harmonized report for all the tools together. I didn't put in the slides the example we ran in Argentina because I thought it's going to be very short on time, but that's how we did. And regarding how easy that is to use, actually in our lab, particularly different members of the lab, one with a lot of genomics experience and one with less genomics experience, run that to make sure we all we can run it at the same time.
Okay, thanks, Josefina. There was a question in the chat and in the Q&A as well about the slides, whether they're going to be shared. Definitely after this meeting, we'll share the recording and the slides. So I don't see any other questions. I'm not too sure with the panelists if there's anyone else who has a question.
I think if not, maybe just to also mention as a result of that question that was asked about running of the programs, I think maybe also just to flag that our infrastructure and pipelines working groups are working hard at packaging these tools so that it becomes easier for people to in fact install these within their labs. And so I think this is something that is quite exciting as we move towards a space where we're wanting to not only standardize our approaches, but standardize our computational environments so that we can ensure reproducibility of the data that gets produced. So at this stage, we're actually well on time. Thank you very much, Josefina. We will proceed to the next presentation. And let me then, at this stage, welcome a good friend, Nikki Mulder, and she is based and she heads up the Computational Biology Division at the University of Cape Town. She is also the PI on the H3A BioNet Consortium, and Nikki is the chair of the training working group within Phage. Nikki, I hand over to you now.
Thank you, Alan. So I will just share my slides. Okay. So thanks, Alan. And there was a discussion at the end of the last talk about training. So hopefully this segues nicely into this talk. So we're talking about the aims and progress of the Training and Workforce Development Working Group. So I am just a co-chair, and Duncan McCannell from the CDC is my other co-chair, and I'm presenting on behalf of the working group members. So the vision of this working group is to develop training standards and resources that will help to create a workforce able to effectively implement pathogen surveillance and epidemiology. So we need to design more general, as Alan mentioned, more general, but also domain-specific and customized training on specific topics. And obviously, these could be multiple different pathogens. Right now, SARS-CoV-2 is the focus, but we need to be able to design this training program for multiple different pathogens and for different stakeholders, so different audiences, from your wet lab to your bioinformatician to public health officials and other government officials. And then we also, one of our other remits is to train external stakeholders on some of the standards and outputs of the Phage consortium itself, so supporting the Phage, the other working groups in Phage. So we decided to think about this in terms of different sort of splits. So there's the training audiences and there's the list of topics. And as I said, the training audiences really are quite a big range of people, from those who are running the sequencing machines and doing the extractions, the epidemiologists who are analyzing data, but also designing studies, the bioinformaticians, and bioinformaticians, there's the data crunchers, so those that just process the data off the machines into a usable format, and those that really analyze and interpret the data. Then we have the healthcare workers and allied public health practitioners and the public health officials who really need to understand the use of, for example, next generation sequencing or other methods in pathogen surveillance and monitoring. And then, of course, we also want to be able to develop a trainer workforce. We want to have enough trainers in these different topic areas trained to have a long-term sustainable training program. And then, of course, there'll be genomics researchers that are interested in analyzing data and learning more and getting trained in this area. So the topics here are just shown according to the workflow of an experiment. But there's a sample collection and, incorporating the Baobab Lens to show how to use that, how to export data, how to standardize the data and use the standards, for example, from the first talk that were described to make sure that these are disseminated and widely used. So there's a really big data management component from the sample collection all the way through to the endpoint and versioning of data and managing of the analysis results. Then, so there's on the wet lab, there's the RNA extraction, library prep, sequencing, and then there's all the dry lab stuff, which includes data integration. So you've got to integrate that clinical data to be able to really interpret the data that's coming out. And then, of course, to generate reports that are usable by the people that are going to implement policies and implement changes based on this data. And then there are various different things available, like protocols, the sequencing platforms need to be taken into consideration, different tools come and go, and we need to stay sort of at the cutting edge of what is the latest tool for what pathogen. So we had, during one of the Phage meetings, we had a brainstorming session in the working group to think about where this group can fit in and what kind of things we can provide. So the first thing is obviously to identify the stakeholders and audiences and to think about the training type and delivery. So some training can be face-to-face, some training can be remote and online. And right now, of course, almost all training has gone virtual. So we sort of thinking about a sort of stakeholder engagement. Once you've got the different audiences that are required to be trained, some of those that were listed on the previous slide, we need to think about what a specific training needs. And of course, everybody comes in from a different background. So, your wet lab person might have done some dabbling in data analysis, or, your bioinformatician might have done some biostatistics or computer science. So you need to sort of think about what is the background of a person, and can you really tailor training to that particular person's needs? But you can do that by thinking about what is a prerequisite for every course that they need to undertake. And then we want to think about building a curriculum. Obviously, there's a generic curriculum. There's certain things that are reasonably generic to multiple different pathogens that you might be focusing on. And then there's the pathogen-specific training curriculum. And then we hope that through this, we'll be able to develop a course plan for each kind of career path. So if you're a bioinformatician and you want to be upskilled in implementing bioinformatics in public health for pathogen surveillance, what is the set of courses that you need to undertake? How do you get there? And what is that career path? Then we also thought about doing the sort of landscaping analysis to see what are existing training courses out there. We know that many people on this in this meeting have been very active in running training courses in the pathogen genomics space. We also have training courses from the manufacturers like Illumina. When they put a machine in, they'll come and do some basic training as well. And so we want to build up this inventory of existing training resources, because of course we don't want to reinvent the wheel if there is existing content or very good courses out there that people can either follow online or actually attend. And then we want to build up a training website with all these resources so that somebody can come and charter this sort of path through a list of available courses. We obviously also need to facilitate training on the Phage outputs, as I've mentioned. Make training materials where there are gaps and align with other relevant training activities. So this shouldn't be done in complete isolation. So we boiled that down into just a set of proposed activities, and I'm going to go through each of these in a bit more detail, but it's basically about maintaining the Phage training related website and putting content on there, developing training resources, that's very broad, collating information on what exists, developing standards for pathogen training as well. We've heard a lot about standards, and Phage is supposed to also be a standards development organization. So this is to maintain, a certain level of quality when you're doing training, and have competencies being given to the people who've actually attended that training, and have they been assessed to show that those competencies have been acquired. But there are also other aspects around standards for training. And then, of course, support the other Phage working groups. So what we've done to date is we have updated the website and done some engagement. We've defined some metadata that needs to be collected when we want to do this sort of landscaping exercise. And then we've developed a survey, and as I said, I'll go through each of these in a bit more detail. So the training and workforce development website, at the moment, it's extremely simple. It's this. Not much more than you see here, really just describing what the remit of the workforce development working group is going to do, and the kind of things we're going to undertake. But the idea here is this will be then expanded to provide a list of training materials, provide user guides through those training materials from both the trainer perspective and from the trainee perspective, to build a community of trainers in the pathogen genomics and epidemiology space, particularly for public health. And also just act as kind of a collator of information without trying to have a copy of everything here. So even if we just have to put pointers to it, we don't want to duplicate what is already existing. It's more a matter of drawing attention to it and putting that together. So we've done some engagement with other stakeholders. We've thought about engaging with the different working groups by meeting with working group leaders. So we had a meeting with the ethics working group leaders, and there we discussed they didn't have any immediate needs for ethics training that they needed from us, but we did discuss how to incorporate ethics and ethics related issues into any proposed training courses that are coming up, or videos or tutorials that are being developed, just to make sure that people are very aware of any ethical issues. We've discussed with the Phage infrastructure working group through various email discussions, and because one of my systems administrators on that working group, there's been they've identified a need for systems administrator training. So that's people who are at the local sites who are able to install software, get the workflows and computing infrastructure up and running for analysis of pathogen genomes. And so in H3 Bionet, we've actually developed sysadmin how-to guides. So we've got different levels of these guides. So really starting from scratch, from setting up a Linux server, building a cluster, And so we're making these available, and these can be adapted. But we've also done some sysadmin training, different levels again, intermediate, beginners and intermediate training. And so we will make all that available. So we integrate, we're working with the infrastructure working group to see how we can support them on that training and building these guides. And then I work with the Africa CDC through the Bill and Melinda Gates Foundation on an NGS Academy. And this is to provide to build a training program with the Africa PGIs, the Pathogen Genomics initiatives, which I'm sure Sophronis will talk about later. And that's also involving Christian Happy's group at HGID. And there's a lot of overlap between what we're going to do, because that is setting up the training for these specific institutes that are building, basically want to implement routine NGS sequencing of pathogens. So I know I've mentioned NGS sequencing and genomics quite a lot, and I realize that there are many other activities around pathogen genomic surveillance. But just to, we aim to cover everything that's relevant to public health. So in the main overlap, in fact, with the NGS Academy has been in working on a landscape analysis, because they also are interested in seeing what training materials are out there. So on that topic, we basically built this sort of inventory. So it's basically a list of metadata that we want to collect about courses. So this on the left, you see some of the fields that we're collecting. It includes important information like who's the target audience. You know, there's academic training versus public health training. Somebody who's going to be doing this for a living versus somebody who's going to be doing this to do some research. What is the format of the training? Is it live? Is it recorded lectures? Is it face-to-face? The duration, how is it assessed? Are these materials available? If you run it regularly, is there scope for other people to join? So you can basically build up a list of not only what courses exist, but whether their training materials are available, whether they're openly available, are they applicable to other people outside of the original audience? And if they plan to run the course regularly, what is the timing of those, and can other people join those courses? So this survey is being developed in REDCap to capture all this information, and that was sent out yesterday to a number of stakeholders. So if you haven't seen it and you're interested in filling that in, we'd really like as much information as possible. And this is completely global. We're not restricting this to any continent. Anybody who does pathogen genomics and epidemiology training should fill this in if you can. And then the aim is to collect this information. At the moment, we've just got a spreadsheet listing the organizations we know of and have been engaging with. So we've had several engagements with people who are training in this space, where the content can be found. But this will, of course, all be pulled together, and hopefully we'll have a nice way to navigate this in a user-friendly manner on the website. So then just a comment now on making training fair. So there are different ways of fair is findable, accessible, interoperable, and reusable. And this helps to keep things standardized, but also to make sure that you have enough information. There's nothing more frustrating than finding a course online and then not being able to track down the materials or who's giving the course or what it was designed for. So the aim of making training fair, and that's what we can implement on the Phage training working group site, is that we want to make sure you can find information about the training and everything you need to know about the training event. So there's this sort of schemas for training instance, and then the schemas for training materials. So are the training materials, the data sets used in that, the VMs that are used for the software, are those all available somewhere? Even if they're not available, at least the information about them is available. So we want to collect the metadata about the courses. So the metadata should be comprehensive. It should describe the objectives, outcomes, audience. And then hopefully there's a link somewhere to those training materials, including information about how to access them, what is the usage, if there are any usage restrictions, and what are the licensing information for that training materials. And in order to do that, we can map to bioschemas, which is, bioschemas is a much broader set of biologically related schemas, but there's a whole subsection within there for training materials. We've actually annotated all our HRA bionet training materials to bioschemas to make them fair. And then, we just hope that this will help to guide users to the most suitable courses and content, so that there's no replication of work. So just to conclude then, the working group is still very new. We only started meeting sort of middle of last year, but we have done some engagement both internally within the Phage working groups and externally with stakeholders. And our working group members include a number of stakeholders from around the world. The survey has been sent out. We hope that that's going to provide a lot of information and also identify gaps where we can maybe work with trainers and content providers to build additional content. We want to help facilitate developing an adoption of science for training in public health space. We want to support training and dissemination of the Phage standards themselves and their other outputs. Obviously, right now, we've identified an urgent need to support SARS-CoV-2 sequencing training. So we're working on a separate effort with Africa CDC on that. But we hope that our activities will be broadly applicable to multiple different pathogens. So I'd like to thank the Phage Secretariat for all their support, the funding from the Bill and Melinda Gates Foundation, and then here the list of working group members. Thanks to them for all their valuable inputs. We've got 2 brand new members just last week, which I didn't get a chance to put in here. But yeah, I want to thank everybody for the input, and thanks to the organizers of this meeting for the opportunity to talk.
Thanks a lot, Nikki. And certainly a lot has gone into the session already. I'm going to just hand over to Ranga to check if there are any questions from the floor.
Thanks, Alan. So we have one question here from Sydney. The Carpentries are also working on creating a directory of community developed training materials. Have you considered collaborating?
Yes, thanks for that question. So we do collaborate with the Carpentries group in PHA4GE. So they have the different sort of types of training of carpentries. There's the programming related ones, there's genomics, there's data carpentry, software carpentry. So what we're doing, the main collaboration, so we've got a whole lot of our people on our network that have attended many of these courses, but we're also working with them now directly to set up a collaboration to build carpentries trainers. And so, so we've had discussions with them about how to build, and this is particularly in Africa, but it could be extended more widely. So I think we'll start with that and see, you know, how we build carpentries trainers in Africa, and then we can start to see examples of where the carpentries practice data sets and their content applies to pathogen genomics. I mean, we personally have not used any of their pathogen genomics related courses because we've used more of the generic R and Python carpentry courses. But yeah, it's a great idea to sort of extend that collaboration and integrate with what they're doing.
Then Nikki, while we wait, while we wait on other questions, I maybe wanted to ask you: has there been some discussion around translation of this material into other languages?
Yeah. So again, not directly on. Actually, we might have mentioned on this group. I don't know. So many meetings. I forget which meeting things came up on. But certainly in PHA4GE we've done that. So we haven't. So what we've done, what we've started doing is transcribing the lectures so that there's subtitles. And that alone helps because we've tested this with sort of French-speaking African countries, and they find that even that helps because if they don't understand the accent of the trainer, they can see it written, and they sometimes understand written English better than they understand spoken English. But the idea is once you've got these transcripts, then it's very easy to do translations. You can plug it into Google Translate. And then we've actually got some colleagues, some in Tunisia that have been volunteering to convert them to basically check the French. And then we've got colleagues in Egypt who are helping to translate into Arabic. So it's definitely something that should be on the cards. It takes money and effort to do that, but it's definitely, I think with the tools available in Zoom now, there's automatic transcribing and things and translating that I think it's definitely a focus area we should consider. You're on mute.
Sorry about that. And if there is somebody on this session now that would like to plug into this, what would be the easiest way to get hold of the training working group?
So we get applications coming in through Nawal from the Secretariat. I think I'll need to go back and check, but I think our email addresses are on the training working group website. But the easiest is to contact the Secretariat, and then they contact us and let us know who's interested in joining the working group. So there are many ways to get involved. You can join the working group and have these active discussions, or we'll probably break the tasks down and have separate task forces to tackle, some of those individual components.
And I think what I like what you were saying earlier about having this collection of training material, because we often find that people tend to redo the material or try to recreate what is already there. So this type of repository of some sort will probably go a long way in terms of just becoming more efficient in how we carry out our training.
Yeah, so that's the idea, is to, if there's really good training material out there, why, develop it again? I find when I'm developing training content, what takes the longest is finding the practice data sets or the examples. And if somebody's already done that, it really does cut down on the time that you take, because they've tried and tested data sets for tutorials and practicals. So yeah, I think it's really valuable to reuse what's there, as long as it's, under an appropriate license that can be reused.
All right, I think we've actually have the emails online, and so I definitely encourage people to plug in. I think that certainly the vision for the consortium is to see how we could stabilize the skills development within our public health institutes particularly, and then more generally within the wider scientific community. And I think a valuable point made on the Q&A around the fact that there are a number of groups that are currently carrying out training, and we should identify what works best and try and partner. Thanks very much, Nikki. Let me just check with Ranga. Is there any other questions that we wish to field at this stage? All right.
Not at the moment.
All right. Thanks very much, Nikki. I am going to then use my powers in chairing this to suggest that we take a 5-minute break. We've been going for at least an hour and a half. So I would like to propose that we take 5 minutes and we then reconvene for what is going to be an exciting 2 sessions. The next one, let me just remind you, is going to be a panel discussion. So with that anticipation, I would then ask that we reconvene in 4 minutes and 30 seconds. Thank you very much. All right, we should all be back. Thank you very much for joining us again. We've got a very exciting talk still lined up for the symposium. And I'm going to ask Noel just to screen share. So everybody, welcome back. It's certainly a pleasure to introduce our next speaker as well. This is Nikki Tiffen, and Nikki Tiffen chairs the Ethics and Data Sharing Working Group of Phage. Nikki's appointment is within the Computational Biology Division at the University of Cape Town, where she is an associate professor in the data integration, as part of the data integration platform of the CIDRA Africa program. Nikki, I'm going to hand over to you. And just to remind the audience that the format of the next session will be a presentation by Nikki, followed by a panel discussion. Thank you, Nikki.
Thank you. Let me just share my screen. There we go. Great. Thanks so much. Thanks for the introduction, Alan, and it's great to see everyone here. So I'm going to present to you some of the work that we've been doing in the Ethics and Data Sharing Working Group. We've entitled the talk, Building a Community of Practice through the Phage Ethics and Data Sharing online platform. And of course, I'm presenting on behalf of everyone in this working group. So the aim or the vision of the Ethics and Data Sharing Working Group is to establish, facilitate, and support an inclusive, diverse, equitable, and collaborative ethics community. So we want to promote ethics research and develop ethics training, but we also want to increase our capacity for having informed, practical, and agile solutions in the ethics and governance space. So data sharing, data governance, sharing biospecimens, they all fall into this space. And I particularly want to flag that we really wanted to make sure that the work we do is around an inclusive and diverse, equitable and collaborative ethics community. So we wanted to make sure that our work is inviting to everyone, that it doesn't feel like only certain people can participate, or that people who are interested are going to struggle to have a voice. So I really hope that we can convince you that, that we've opened that space successfully. So what we realized is that most of us, and we discussed this quite a bit, most of us are what I would call applied ethicists. We don't have formal ethics training. We're not dedicated bioethicists. We're actually often more data-driven or biospecimen-driven researchers who are trying our best to undertake our research or to promote research, but to do it in a way that's ethical, upholds ethical principles. To make sure that our data sharing is done in the most ethically suitable way. So we need practical and applied solutions. And this is what we decided in the working group, that we really wanted to make sure what we produce from this working group is stuff that can be used and that is easy to use and can be relied on to make sure we're ethically compliant without us having to be the bioethicists necessarily. So we discussed whether we need more guidelines and recommendations, and we realized that there really are quite a lot of guidelines and recommendations out there. There are a lot of different working groups that have done great work in this space. And if we were to do guidelines and recommendations in the working group, we're not quite sure how we prioritize where we should start. What would be the most important? Who would decide what's the most important? And the more we discuss these kind of things, the more we've realized that what we really want to do is to build what we've called a community of practice. So we work out what the current issues are by engaging with a community of people working in the field who are struggling with certain things. And that way we can find out where we need to direct our efforts from the people who are actually on the ground trying to do research in an ethical way. And then if we can organically identify these kind of current issues that are surfacing in a kind of bottom-up problem-solving way, then we can also organically develop solutions that are appropriate, that are practical, and that can actually work effectively for the people who need to use them. The other thing that we've noticed a lot, there are a lot of really great working groups in the kind of LC space, the data sharing space, governance space, but we're not very good at talking to each other outside these specific silos for each of the working groups. And in fact, just to add one of, which I won't be talking about further here, but one of our plans is to actually try and convene a meeting of working groups in the LC space to start to work at talking across the different working groups and sharing our wider network of skills and knowledge. But we also wanted to develop something within our working group space that can really help to promote the conversation in this wider network of people who have skills and knowledge. And we have such varied skills and knowledge. We have the pathogen space, human genomics space. We have, you know, kind of building data tools, people who work in health informatics and work with electronic health records. And all of these bring a different lens onto the ethics and data sharing issues that are coming up. So the question is, how are we going to bring this all together, and how are we going to learn from each other and build a collective approach and a collective solution to doing better ethical research and better ethical data sharing with kind of manageable solutions that are easy to follow and easy to implement. So we came up with a plan to build the Phage ethics and data sharing platform. So this is a simple browser-based online discussion platform. There's nothing elaborate or complicated about it. You don't need to download any apps. You won't be bombarded by notifications. It's kind of a discussion forum in a fairly vanilla discussion forum kind of structure. But the idea is that it'll be open to everyone who's interested in anything to do with ethics and data sharing. And it's now live, the platform is live, so you're very welcome to log in and have a look. Hopefully not everyone will log in at exactly the same moment and put us under our first ethics and data sharing platform stress. But we decided that it would be open to everyone, but we would request people to create accounts to participate, and this is just and to identify ourselves as members. And this is also to help with building the community of practice, where we all know who we're discussing with, who we're talking to, who we're sharing ideas with, and then obviously, of course, just to protect us a little bit from you know, floods of trolling from the outside world who aren't really interested in ethics and data sharing. So the aim of the forum is that it's a space to discuss and problem solve current ethics and data sharing challenges, and to share opportunities, to collaborate, to build collaborations, and to kind of break down the silos. So there's no hierarchy. If you have an idea, you can get on the platform and put it out there. You don't have to be, you know, a chair of a working group or in a specific kind of organization in order to be able to get talking about these things that we're all concerned about in the ethics and data sharing space. And it also recognizes that we're not all bioethicists. Some of us are researchers just looking for solutions to particular problems that we have at hand. So this is kind of the landing page for the platform. Don't worry too much about the very small print, which I can imagine is not so great on some of your Zoom screens. But of course, if you go to the platform, you'll be able to see these different kind of topics that are here. There's an open discussion. So this is like a very typical landing page. You can click on the little logo that says Ethics and Data Sharing Community, and that will always bring you back to this landing page. And we have different categories. They couldn't all fit on my screen grab, but I'll just talk you through some of the interesting ones that we have. So the ethics and data sharing category, if you click on that, this is where we envisage most of the discussions and the research collaborations taking place. And it's also a place where if you have a question, burning question, you can put it out there and see what the ethics and data sharing community have to say about it and start some kind of discussion. So to go to that space, you literally just click on the category on the landing page, and you'll go in and you can see these different threads. So as a working group, we've all got some projects that we would love to see implemented, and we maybe haven't had the time or the right teams to do it. So we've pre-populated some of these. just to get us going, and because we'd love to see the work done. So you can participate in an existing thread. You can just click on the thread and it'll take you through to a detail of what that thread is about and who's asking what. And then within the thread, you can reply and get involved in a discussion. So some of these are discussions which are just general, let's talk about this. I've posted a couple and we'll come to more details. There's a systematic review I'd love to see written, some more kind of meaty projects. So it's really open to you. Do you just want to ask a question, or do you want to build a like 3-year collaboration that's going to keep you all busy for a long time? If you have something new, then you can start a new conversation, and that will build a thread here that you'll become the owner of. So please, everyone, we're inviting you to start something new. Jump in if you've got a burning ethics or data sharing question and you'd like to build it into a discussion or a project or something like that. Here's your opportunity to start a new conversation in the ethics and data sharing space here. We also have, as one of our categories, a section on funding. So this is where you can post information about funding opportunities that maybe your organization is offering, or that you know about, or you maybe would like to just share with everyone. But we also will put some information here about funding opportunities that become available through Phage. And we have some Phage small grants available that we have just recently posted on here. So I invite you all to have a look at that call. You can also, so this is what it looks like. You can click on the funding and come through to this particular thread. And here we've posted all the information about these small grants. So the small grants are for short-term projects that are low cost. And there are all sorts of suggestions here, what kind of projects. Maybe you want to write a systematic review or scoping paper, and you want to reimburse your colleagues who spend time on this, or you want to build a framework or run an online workshop. So please do have a look. There's a link to the application form. It's not too onerous at all. We made sure it was quite short and sweet. And to the point. So we're hoping to get some great applications from you all through this space here. We've also got some other categories. If you've read an interesting paper, post a link to it here and we can have a discussion around it. New publications that you've got out that you'd like to share with everyone. There's a training and courses section, and in fact there is already a post in the training and courses section for a training opportunity that's become available. So you can offer training, you can collaborate together, put a team together when you want to produce some training, but you can also put a call out there if there's some kind of training that you desperately need and you would like to see that training happen. This is a space to see if other people agree with you and to refine exactly what you're asking for. And then, as you heard in the earlier talk from Nikki, that we can discuss how we can fulfill training needs as they go, and there may be other people who are also posting who can offer the kind of trainings that you're looking for. And then we know also a lot of researchers who have to do IRB applications can get really caught up in those, and they can be quite tricky. So we made a special space here. If you have a question about your application, or you got stuck, or you don't know how to take it forward, or you don't know how to approach your ethics applications for your review board, you can actually put a question out to the community here and see if you can get some advice from the general ethics community and get some helpful hints. And then obviously any specific Phage announcements and projects, we also have a space where we'll post them. So I just want to recognize that the team, we're quite a small team still, but we're hoping through activities on the platform that we'll be able to encourage more of you. People who are very interested and active may want to get more involved in the working group. So I'm the chair, my co-chair is Anya Badeka, and these are our current active members. And then I'd also just like to thank the Phage Secretariat who've been supporting the working group, and particularly Alicia Naidu, who's been really helping keep us on track, and Jamie Southgate, who's done tremendous work in putting together the forum for us and helping us with all the documentation behind that. And then also just to recognize that the Phage Ethics and Data Sharing online platform was created using funds from the Phage funding provided by the Bill and Melinda Gates Foundation. So what I'm going to do now, we're going to have a panel discussion. So I'd like to introduce the 3 extra panelists who are going to join us. Well, I say I'm going to introduce them. I'm actually going to ask them to introduce themselves. And we're going to give you a sneak peek at some of the posts that we've already put on the platform. So the idea is just to give you an idea of the kind of things that we've put up there. But of course, we're really hoping that as a community, everyone will step up and get involved and put your own questions up there. So we're really, really looking forward to seeing the kind of inputs and the kind of things that people would like to talk about on there. And maybe you've got your own projects that you would like to put out there for collaboration. So what I will do, and then after I've Alice, Anya, and Brianna have just told you a little bit about their projects, and I'll tell you about 1 or 2 of mine. Then we'll open the floor for comments, questions, and feedback. So I'm going to hand over, first of all, to Alice. Sorry, we seem to have some feedback from someone else's mic. Alice, can I hand over to you? And you can introduce yourself and tell us more about the one or two of the projects you've posted.
Thank you, Nikki. So I'm Alice Matimba. I'm from the Wellcome Connecting Science, and I develop training in genomics and bioinformatics. But I've been involved in ethics research for some time, and quite interested in developing tools that are useful for researchers. And so here I have posted 2 projects that could be interesting to people. So it's still early stages, particularly the first one, where just recognizing that there's a lot of data in ethics that is being generated, and obviously a lot of interesting questions all over, about ethics in genomics, or ethics tools in genomics. And just like you have a lot of data, being archived from genomic studies, this is not really the case for ethics data. And yet there's a lot of information that could be useful for researchers. So we're proposing developing a database that could be useful for people to interrogate across different research projects, what data sets are available, identify what studies have been done, what questions have been asked, and particularly just knowing that ethics is, it's regional dependent, cultural dependent. So it will be important to be able to interrogate those kinds of questions. Which populations have been studied and what have been the results? And so the first step would be to try and develop a standard, a metadata standard, so that we can be able to then curate information that's coming from ethics research projects. The second project that I'd like to share with you is on developing an ethics and governance framework for translation and commercialization of human genomic data. And so I think a lot of researchers are now finding themselves in a situation where they need to understand what they need to put in place in order to prepare for a situation whereby their data or their research may end up being commercialized, and thinking around those issues. So we're trying to come up with a framework or as a guide for people to think around what type of, do we need to have people consenting for this specific part of the research and this part of the translation of their research, and what type of information do we need to provide to participants in order for them to have the right consent and to have the right tools for them to be able to commercialize any of the genomics data. So these are 2 projects that we're looking forward to having people on board. So if you go onto the website where which Nikki has mentioned, you'll be able to click on there and respond, and we can start having a conversation around these topics. Thank you.
Wonderful, thanks, Alice. These are very exciting projects, actually, and yeah, interesting and new. So I'm very much looking forward to seeing them also moving forward. So let's see, now we have, so yeah, Brianna, I'm going to mute myself and hand over to you, and you can tell us a bit about your projects, a couple of your projects that you've also put on the platform. Thank you.
Yeah, thanks, Nikki. So hi, everyone. I'm Brianna Lindsay. I am an epidemiologist. I am affiliated with the University of Maryland, Baltimore. I'm currently working in Lusaka, Zambia, on with CDC and MOH on HIV prevention and treatment programs. So my interests really lie around data sharing experiences. So I've proposed a couple of discussions to take place. However, I'm obviously open to seeing where these discussions go and what types of projects could potentially come out of them. I think a number of the other presenters have kind of made some references to some of the experiences that we have with genomic data sharing, particularly within the past year, where this has been increasingly kind of pushed to the forefront and very important. One of the topics that I had to discuss potentially is how to share successful genomic data sharing experiences. So what lessons can we learn to take things forward? I think sometimes people can be a bit timid about initiating genomic data sharing. I think particularly with sensitivities around metadata. And I think that any discussion that we can have around what are some of the examples of best practices. I think we have a lot of these examples in human subjects research for ethical data sharing, but now that genomics has come so far, we can see how that will be applied to genomic data sharing moving forward. We certainly have some experiences that we've learned from the COVID space as well. So I think that's looking forward to participating in that discussion. And then the second topic I had really was around establishing a global principle to harmonize disparities in data sharing guidance. So I think, or if there is one that exists to point people in the right direction, I think there are many countries and many different institutes and many different organizations have a lot of different guidance around data sharing. And I think it would be great to kind of provide a reference to any sort of global principle or think about how we can establish what are the underlying principles that constitute a good data sharing guidance. And so I also just pulled out this very scientific PubMed graph on genomic data sharing. And as you can see, it looks like we've kind of reached our peak in the early 2010s. So we'll see how genomic data sharing guidance and publication happens moving forward. But I think these are important things that we can definitely discuss using the platform, maybe, and formalize into more collaborative projects. So I look forward to kind of talking to everyone in the forum. And thanks, Nikki, for allowing me to promote these discussions.
Thanks, Brianna. No, it's a pleasure to have these things all put out there, and I'm really hoping that everyone will jump in and share their opinions on these different things, and share, as you say, some of sharing when it went right instead of always worrying about when it went wrong. I think will be a really interesting discussion to follow up on. So let's Anya, so I'm going to hand over to Anya to introduce herself, and she can flag for us one of her discussion points that she's also put into the open discussion. So Anya, I'm going to unmute and hand over to you. Thanks.
Thanks, Nikki. My name is Anya. I'm a research associate at the South African National Bioinformatics Institute. I'm a research psychologist, so focus not that much on genomics, but more the ethics behind it. So recently in South Africa, we've had the Protection of Personal Information Act coming into fruition. And then also you have the EU's General Data Protection Regulation. And what's interesting about it is that those regulations do not apply to anonymized data, which obviously has a significant impact, because that means that anonymized data can be used for longer periods of time and can also be used for purposes for which it wasn't originally collected. The problem is that anonymized data means that it's irreversibly de-identified. And the problem with genomic data is it actually possible that genomic data can ever really be anonymized? So I put that question up on the discussion group. I hope you guys will join and give your opinions on it, because it's purely a question that I'm very interested in finding out. Thank you, Nikki.
Wonderful, thanks, Anya. I think people who've heard me talk before know that I also bring this point up very often about the fact that we can't anonymize data. So I'm hoping that we'll have a lively discussion in that space. And if we can't anonymize data, how are we going to make sure we can share those data safely? So I'd like to, at this point, I'll just go back to the page with the with our panelists and ask if there are any questions and who would like to, I'm just looking, sorry, I keep looking to the right because I've got two screens here. People who would like to bring discussions or suggestions or questions that we can answer. And I'm also conscious of the time because we are kind of running a little bit behind schedule now. So Alan, I will also look to you to rein us in if need be.
I will certainly do that. We've still got at least 3 minutes, so you are still on target.
Okay, great. Do we have any questions from across the board? Or not so many at this point?
Hi, Nikki, this is Nikki.
Hi.
Hi. I don't know if you've mentioned it at all, but are you guys looking at the sort of application of the Nagoya protocol in this space? Does it, I would imagine it, they're trying to extend that now to genomics data. So how does that apply here? And is it something that you've thought about?
Yeah, that's a great question. I actually realized that I forgot to present my couple of questions or projects that I had advertised. They were on the last slide. So you'll all have to just go online and see which projects I've got going. So yeah, I'm very interested in the space in benefit sharing. And as you know, that's something that I've already published on. And I'm interested in building a kind of benefit sharing platform or a framework that will address some of these questions. I mean, the Nagoya protocol currently is not for human data at all. So there's this space about negotiating how we do benefit sharing. We've got to incorporate the Nagoya protocol for things like pathogen samples. And the other thing about the Nagoya protocol, of course, is that it doesn't yet cover the sequence information. It only covers the samples. So one of the projects on building this benefit sharing kind of, I'd say, framework, is to work out how we can take the principles that underlie benefit sharing, like in the Nagoya protocol, like in kind of community engagement approaches, and how we're going to draw those together into a framework that if I'm a researcher setting up my protocol, I can very quickly hook into how I can effectively look at benefit sharing or maybe incorporate it into my program, right from the beginning. So it's very definitely an essential part of what we're doing in the ethics space, and certainly something, there is a project already loaded up and ready to go. So I don't know, other panelists, if you would like to comment on that or add further. And again, Alan, I'm trusting on you. I'll just put my slide up that I forgot to talk about.
Just before your panelists answer, just to let you know, we have a few questions from the floor as well. So we will raise that as soon as you're ready.
Okay, can I just jump in? I can see the questions. And then panelists, how will you just unmute if you want to answer any of them? So the first question is from Julia. What lessons are there that can be learned that would apply to other health data sets beyond genomics? So I will quickly answer from my perspective because I also work with electronic health records. And I think that the lessons are very generalizable. So we have genomics, which is genomics for human data. We have genomics for non-human data and pathogen data. But then we also have the whole clinical phenotype kind of set up with those data. And I think that these lessons have to be generalizable if they're going to be of use to the community. Other panelists, would anybody else like to jump in there?
Yeah, I think Nikki, I think my response also kind of overlaps with the second question that's also in the Q&A, in that I do think that building these examples of when things have gone well and things have gone smoothly are a good way to kind of demonstrate how data sharing can be ethical and really straightforward in some cases. So I think providing examples to people will always kind of give them comfort. And I think that that would be a way to kind of expand the conversation. If we can build this repertoire of kind of good examples of data sharing and ethical work.
Sorry, Nikki, could I maybe just alert you to we have one person's hand up on the floor, and maybe I could direct that person to ask their question at this time.
Yes, absolutely.
Hello.
Hi.
Yes, sir. Carry on.
Hello, Nikki. Thank you. I just want to ask. I'm Yomi from Nigeria. I want to ask opportunities that are available for training of early research careers on ethics, and two, can you also share the link that you discussed on during your slide presentation? Thank you.
Okay, great. No, those are both great questions. So can I ask Jamie or someone to just share the link in the chat to the platform? And then, yeah, so if you log onto the platform and create an account, which is quick and easy to do, then you can go to that training session, category from the main page, and that will, you'll be able to read there whatever trainings come in. But I think even more importantly, I mean, that's a great place if there are particular kinds of training that you'd like to have in ethics, is to start a discussion, start a new conversation under the training section and let us know what kind of ethics training you're after. And then we can start to look at resources and have a wider discussion and see how we can work to pull those elements together and provide the kind of training that's needed. So I'm hoping that, Jamie shared in the chat, again, the link to the platform. And we'd really love it if you actually come onto the platform and we can dig in more deeply and find out what kind of training you're after. It would be wonderful to see you on there. Thanks. Shall I jump to these questions from Peter?
Let's take one more, sure.
One more, okay. So Peter said, we have seen a reluctance to share pathogen data during this pandemic, with data often spending weeks or months on the desk of people in health departments. Some of this seems to be due to the novelty of both sequence data and also data sharing in health departments. As Phage, do we risk preaching to the converted? How can we bring more DOH people into these discussions? So this is a great question, Peter. And in the interest of time, I'm going to ask you to come onto our ethics and data sharing platform and post it there as a new thread. And again, let's get everyone's input. And I guess, Julia, we don't have time to address your follow-on question, but these are all the kind of questions we'd like to dig into there and involve a wider community in problem solving. So yeah, that's my kind of overarching, rather boring, repetitive answer is see you on the platform. Thank you.
Nikki, could I maybe ask that you just deal with that last question of Julie, and then we will move.
Okay,
then we'll
sure.
move on to the next one.
Okay, so the question is, what more can we do to encourage the research community to share data in a timely fashion? So I don't want to be the only one talking, so I'm first going to offer if any of my panelists would like to jump in there. Otherwise, I'm happy to give it a stab. I don't want to put anyone on the spot, but it would be great if you, yeah?
Nikki, I'd like to chime in. I think quite often what happens within the scientific field is people tend to hog their own data. People become extremely overprotective of it, and it's my data, I collected it, so only I will use it. And the thing that people need to remember is that that data, the more it can be used, the more people it can help. So therefore, it's an ethical requirement, in my opinion, to share that data with as many people as possible, because that's going to make what we gain from the data more valuable. So, well, that's my opinion.
Yeah, I agree with you, Anya, that we all desperately want to promote sharing of data. I think it's quite a tricky question to answer, Julia, and I think maybe one of the things that we need to start with is to ask why people don't want to share. And I agree with Anya, in many cases that's because people want to do the research on it themselves. But maybe we need to step back and also dig a little bit deeper and see if there's some kind of concerns or fears that we can address to help data sharing feel less threatening to researchers. So that would be, yeah, my addition as well. Yeah.
I think, Nikki, while we transition the slides, I would like to give an opportunity to Maritha Kotze, who has a hand up, and maybe we could field that question as we change the slides.
Thank you for the opportunity. It was a wonderful, presentations from everybody, and I just think that what you are touching on and what you're trying to do is wonderful. I think in a way I've been trying a lot of these things in a way on my own, and trying to finding the right frameworks and ways to work on the ethical side. But what I find is that there's lots of obstacles, to share data. Starting just with having to do the material transfer agreements, but then it changed into data sharing agreements, and many of our projects then is about both material and data sharing. But what I haven't been able to even accomplish within my own university, and I'm not blaming anybody, it's just that there is so much, uncertainties between different people that do not understand why do you want to share data. So just trying to get an inter-university data sharing agreement, it is very difficult. If you share data between one university and another, where the collaborators or the investigators are part of a research project with ethics approval, with everybody having gone through the investigator declaration, signed, that they read the protocol, provide their CVs, you submit it for ethics. But at the moment, you want to now include more people, then the time that it takes to get everything signed, and all the inter-universities, all signing this data sharing agreement. This is definitely something that needs to be discussed, and I will also try to find time, to interact on your forum. So is there anybody else that experienced this type of, problem to have a data sharing agreement signed? Because maybe we need a template for that, so that maybe even once I got mine sorted out, it could be something that can be shared on the platform if the university would allow. But everything is about, you can't just do anything without getting the approvals from the legal people, from the ethics people, and from your committees.
So Marie, let's turn that into a discussion. I think that would be a really helpful discussion, and even try and get some university reps to come onto the platform and also help us there. I think that's a great opportunity to take that forward.
Okay, thanks. I'm actually in the process working with one of our legal people, and I have to reply back on a document that needs to be signed, and all these things actually comes up. And it feels sometimes, like you're the only person trying to get this through. Is there not anybody else in the university that's trying to share data? That kind of thing that you sort of experience. So I will definitely follow this up. The first thing I'm going to do is then I have to reply anyway to this, for this document that I need to try and get approved. So I will actually...
And we'll draw on your experience in the space.
Yeah, I will use some of it.
You can blaze the path for us, yeah.
I'll try and help making a path, but I'm also very interested and just want to say in front of everybody that last slide that you showed to invite people on your article, I would really like to collaborate. We have published together in the past, and also we have created a new theme for Frontiers in Genetics, where the title of that topic is Implementation of Genomic Medicine in Africa, One Continent, One Vision. If anybody is interested in that, I would like to extend that invitation. I think, Nikki, I did send an invitation to you.
Sorry, Marita, could I maybe just interject quickly? I wonder if we could post that information on the forum. It might be useful for the much more wider community. And I think maybe at that stage, we should maybe just transition to the next talk. I think we've had quite a vibrant discussion on this topic. And just looking at the chat, there's quite a few people that are certainly going to be joining the working group. And I think we are probably all encountering very similar challenges. And I think that having the space in which to try and share some of our experiences would actually go a long way in trying and work through some of these. Thanks very much, Nikki, and the panel.
Yeah, thanks, panel, as well. And thanks, everyone, for listening. And we'll see you online. Bye-bye.
Thank you. So with that, I wish to move to our last presentation for the day, and that is by Sophonius Tessima, and he is an implementation science specialist at the Africa CDC, and he's going to tell us about the continental response that the Africa CDC had done in response to the COVID-19 pandemic. Over to you, Sophonius.
Thank you, Alan. Thank you, the organizers, for inviting Africa CDC to present the work in progress in supporting SARS-CoV-2 sequencing in the continent. If I share my screen. So my name is Sophonius. I work at the Institute of Pathogen Genomics at the Africa Center for Disease Control at the headquarters in Addis Ababa, Ethiopia. So I'll give you a very brief update what pathogen genomics, how we understand pathogen genomics at the Africa CDC, and what are the updates on COVID-19 sequencing efforts. And then I'll summarize my presentation. But before I go into the details of my presentation, I would like to congratulate the organizer for such a diverse and very useful discussions and presentations today. And all the technical working groups of Phage with all the progress that we've seen today. I don't think I should talk about what is the benefit of pathogen genomics for this audience. It's transforming public health across the world, but these potentials of pathogen genomics haven't been yet fully harnessed in the public health space in Africa. Africa CDC started with the question that what are the challenges of implementing public health pathogen genomics in the continent? Together with, in collaboration with African Society of Laboratory Medicine and SANBI with Professor Allen's group, we've conducted a series of assessment of genomics and bioinformatics capacity from 2018 to 2020, which included on-site visits to 22 institutions in 11 countries, virtual assessments, and data from different sources. All this data is summarized in a very high level into 2 slides. The first finding is, as you can see in the continent, we've seen some capacity in terms of NGS equipment. What this map shows, the bubble size is proportional to the number of NGS devices that are available in that country. And then we asked the question, how much of these NGS equipments are in public health institutions? So what we found is more than 80% of these equipments are not in public health institutes or are not in institutes that have some sort of affiliation with public health institutions in the continent. This and the detail of this report is published recently on the Lancet Infectious Diseases for more details. The other findings are a lack of coordination across the continent in terms of developing guidances, policies to promote, to enable pathogen genomics implementation. Clearly, there is limited capacity in terms of bioinformatics. There is a severely inadequate technical workforce on public health pathogen genomics, public health laboratory technicians, as well as bioinformatics, molecular epidemiologists, and others. And there is a limited translation for public health decision making as well. So in response to this assessment and challenge in the continent, Africa CDC established the Institute of Pathogen Genomics in November 2019, with a vision to integrate pathogen genomics and bioinformatics into public health surveillance, outbreak investigation, and improved disease control and prevention across the continent. The institution is tasked with 5 cross-cutting priorities: creating enabling mechanisms, policies, and guidelines, strengthening capacity, establishing a pathogen genomics and bioinformatics network, and creating more functional data systems and implementation of priority public health use cases. Since the launch of the Institute on November 2019, after a month or two, COVID-19 outbreak was reported. So that changed the directions and the activities of the Institute to focus on supporting COVID-19 sequencing in the continent. Early on, we've been advocating for the need to accelerate genomic-based surveillance for the continent to strengthen, to support the outbreak response in Africa. So since March 2020, Africa CDC has been supporting, together with partners, lots of partners, private, public, philanthropic partners, supported more than 16 countries in the continent with sequencing equipments, reagents, training and mentorships and technical support, as well as series of webinars in terms of building a community which can share resources, protocols, and expertise. In our efforts, we were able to distribute 1 NextSeq for MySeq, 3 bioanalyzers, and 10 MinION MK1B devices and the like. This effort in terms of supporting member states for COVID-19 sequencing actually grew into a much more bigger initiative, which was launched in October 2020, the Africa Pathogen Genomics Initiative. This was launched in partnership with the African Society of Laboratory Medicine, the Bill and Melinda Gates Foundation, Illumina, US CDC, Microsoft, Nanopore, and many other additional partners. The initiative is focusing on key components, strengthening the Institute of Pathogen Genomics within the Africa CDC, so that we have the capacity to coordinate and mobilize resources to create enabling mechanisms, policies, and guidelines, as well as to build the community, pathogen genomics community of practice and technical working groups to support and enable pathogen genomics across the continent. The second priority is pathogen genomics and bioinformatics network. In the continent, there are several networks, but how do we bring these different silos networks into a more broader pathogen genomics and bioinformatics network, which involves capacity building in national public health institutes. The third component is data and data architecture and systems. How do we ensure that we have a continental data architecture and data system that works for the public health institutes in the continent? The fourth component is the NGS Academy, which focuses on workforce development in genomics, bioinformatics, genomic and genomic epidemiology. The fifth is implementation of high impact genomic use cases. So all these different components are progressing together with, again, several partners that I'm not listing here. As part of this initiative and in collaboration with the World Health Organization in Africa, we created a sequencing network for COVID-19 and other emerging pathogens. And this network has 3 layers: the specialized genomics and bioinformatics centers, that the ones that you see at the top. We have the ASEGIT in Nigeria, CRISP in South Africa, and SANBI for bioinformatics in South Africa. And there are regional laboratories covering all the different regions of the continent. And then there are national level COVID-19 sequencing laboratories. So this doesn't mean that in the national levels are not generating sequences. There are labs that are generating lots of sequences from national level labs. There are labs at the regional level that still have limited outputs in terms of sequencing. But this sequence was generated with a view that how do we bring together the continent in terms of sharing pathogen genomics platforms, resources, based on the existing structures, based on representation of all regions, and based on equity for access to sequencing facilities. So we prioritized the operationalization of this network to accelerate SARS-CoV-2 sequencing in the continent. Our focus is really to leverage the existing capacity and coordinate the sample referral process across this network to ensure access to sequencing facilities to member states that have no or limited sequencing at this stage. And then we are also encouraging and working towards more improved data sharing across the continent and with the global community. Our plan in the acceleration of SARS-CoV-2 sequencing is to reach to 50,000 SARS-CoV-2 genomes by 2021. When we developed this plan, the continent was at around 5,000, 6,000. Currently, we are at 10,000, but it doesn't mean that all sequences are generated as part of support from this network. So this map shows the different regional labs that are currently supporting member states that have limited or no sequencing capacity, in addition to supporting their own countries. We have institutions across the different regions of the continent, and these institutions are currently supporting their country plus at least 3 countries in their region. So our approach is to leverage on the existing capacities in these institutes and to start simple and slowly establish a routine surveillance system to support the sample collection and shipment logistics, to support the regional hubs in terms of personnel, training, sequencing reagents, as well as possibility for equipment upgrades to increase throughput. And the last area of support that we are focusing on is data analysis, sharing, interpretation, and reporting. How we see this in the operationalization of this network, we see that at the member state level, we have there are things that needs to be done in terms of putting a good sampling framework to identify what to sequence. Good quality samples should be collected and properly stored, and collection of complete metadata is critical. So Africa CDC is working in terms of engagement with the countries, with the member states, to address these gaps at member state level. At regional sequencing hubs, as I said earlier, we're trying to support personnel and training needs. The training aspect is being done in collaboration with H3A Bionet. Reagents and supplies, quality control at the lab level to make sure that good quality sequences are generated. Equipment upgrades and possibility for automation to increase throughput, to minimize the burden in these regional hubs, to support other member states that have limited sequencing capacity. But in this loop, we see there are challenges with the specimen referral. Africa CDC, together with partners, are trying to formalize the material transfer agreement, to address challenges that are related to import permits, attempts to standardize and complete, making sure that complete metadata is collected and shared with the regional hubs so that it is linked with the genomes, sample packaging and shipment, as well as how do we move the samples from one country to the regional hub, which involves courier service as well as challenges that are related to custom clearance. We see challenges that are related to data standards and management, turnaround time, how quickly we can get the data from the regional hub to the countries, standardized reporting mechanisms, and ensuring data is shared and used to inform the outbreak response. So Africa CDC, through its Africa PHA4GE initiative, is working towards closing this loop. So in doing so, what are we doing so far? There are 12 specialized and regional hubs. We are allocating about 2 million in civil work to support personnel and other immediate needs in these regional hubs. In partnership with Illumina and Oxford Nanopore Technologies and other partners, we are distributing about reagents that's sufficient for more than 25,000 genomes. We are in discussion with lots of partners to work towards upgrading equipments and automating labs. In collaboration with H3A Bionet, a training need assessment survey is being conducted. Once that's finalized, we will have a training for these regional hubs. In sample referral process, we put together an MTA in both English and French that can be used by the regional hubs. And we have a courier service in place that is able to send packaging materials, dry ice to the countries and send back the samples to the lab so that the samples are not compromised during the shipment process. In this sample referral process, 3,700 samples are already referred from 21 countries, and 16 of these countries have received their results so far. Of these 16 countries, 13 of them have reported variants of concern. So, so far, I think as of this is updated on the 29th of March, 9,700 genomes are currently reported from 36 countries. And 18 member states are reporting the B1351 variant, and 18 member states are reporting the B117 variant, and 9 countries are reporting both. So while we are trying to operationalize this network, there are several challenges. One is mainly related to the limited sequencing and bioinformatics capacity in public health institutions. But there are success stories. Some countries, some national institutions are able to show that, showcase that this COVID-19 can be an opportunity to establish genomics capacity locally. We have some examples at national level laboratories. For example, Zambia is a good example in local sequencing. But we are trying to support as much as we can to all member states to have the member states that have some sort of some level of local capacity to strengthen that and produce sequences locally based on the needs of the local epidemiologic setting. The lack of standardized sampling framework, which is tailored to the African epidemiologic setting, is not there. So we are working with partners in WHO to establish a sampling framework guide. Hands-on trainings are needed at regional and national laboratories. That has been very difficult due to the COVID-19 travel restrictions. Lengthy process for approvals, material transfer agreements, and import permits is another challenge that we are trying to address. And there are also logistic and custom challenges and standardization that's related to metadata, protocols, data analysis, and also reporting frameworks. So we are trying to work with different partners to address each of these challenges. In summary of my presentation, acceleration of SARS-CoV-2 genomics is needed in the continent, especially timely data generation, analysis, and sharing is required to monitor the variants that we know now and the emergence of new variants. Since we are seeing some third wave of infection in some countries across the continent and the slow vaccine rollout in Africa. So this probably urges the need to accelerate SARS-CoV-2 sequencing in the continent. The other message that I want to deliver today is Africa PGI is a platform for partnerships, collaborations, and coordination to strengthen public health pathogen genomics and bioinformatics across Africa. So the initiative is not only focused on COVID, we have lots of other priorities, but currently based on the capacity that we have, we are focusing mainly on COVID-19 sequencing. So this is what I have for today. What we are working on is only possible because of the support from lots of partners. Bill and Melinda Gates Foundation, Illumina, the US CDC, Nanopore are currently really supporting our initiative with training, the Chan Zuckerberg Biohub, Fogarty, Johns Hopkins, H3A, Bionet, Sanbi, are supporting us. And as you can see here, all the regional hubs that are working tirelessly to support their own country as well as other countries that have limited sequencing. There is a long way to go. There is an ambitious target that we are trying to put together, but together with these partnerships and collaborations, with the spirit that we can create a functional and operational network in the continent, we think we can achieve that. So this is what I have for today for this update, and thank you very much for listening to me. Thank you.
Thanks very much, Sophonius. And we could take a few questions, and if you have a question, you could either just raise your hand and we will unmute your mic. Alternatively, you could just post the question into the Q&A. And maybe, Sophonius, you know, as you were speaking, one just got a sense that you've got quite a few questions to add to the ethics working group's platform, and I think particularly of the material transfer agreements, among other things. And I was just wondering, in your opinion, would it be feasible if one now, or at least now that you are finalizing a material transfer agreement, to have some reflection downstream once people have used it to try and assess just how well it worked or how well it did not work? And I'm particularly thinking of many of us who are challenged with getting a material transfer agreement within our own city, let alone across borders.
Yeah, thanks, Alan. So the material transfer agreement component is what we are trying to do now is a kind of emergency use material transfer agreement rather than a very complete, well-vetted, well-discussed and scrutinized material transfer agreement. We are using this as an emergency use. We know we have received comments from different countries, but we are collecting all those informations, and in the future we will have much more, detailed consultative discussions to really improve that material transfer agreement, and also cover beyond COVID, so that we have more comprehensive and broader material transfer agreements in place with the countries, so that next time when we have another emerging pathogens, we don't go into the same route of going through the same material transfer agreement. So this is a work in progress, and we are considering it as a somehow emergency use material transfer agreement. There are lots of things to be improved on it.
Just while we wait on another question, I just had one more question there from my side. And, pre-COVID-19, there was always the logistical nightmare to move or to ship reagents across the African continent. And that was in part a systemic issue as well, with not having sufficient either sales reps from a certain company. But I'm just wondering, as a result of responding to this pandemic, do you see the repairs or the solutions becoming one in which companies are coming on board to look at more easy logistical ways of actually improving this? Or are we going to be at a point where we actually need this continental-wide push each time we have a crisis? I wonder if you had any sense from the discussions happening across the continent whether we are moving towards a solution that is going to actually deal with these systematic problems versus just a response to the need.
Yeah, thanks, Alan, for bringing that very important issue. I think all of these are components of the discussion of partnerships with the Africa PGI. When we have discussions with Illumina or discussions with Oxford Nanopore Technologies or Thermo Scientific, this is one of the main things that we discuss: access to their reagents, logistical challenge, technical support, training. Without this, it would be very difficult to implement public health pathogen genomics in the continent. There are still challenges, but we've seen a very, very huge improvement. I can give a simple example. Before the COVID-19 outbreak, there was one supplier or channel partner for Illumina, which was based in Dubai and serving 54 African countries. But today that reality has changed and there are more than 20 channel partners in the continent for Illumina. So these are the kind of things that the private companies are realizing and working with us, working closely with the Africa PGI partners to address this challenge. So we expect more from the private companies to address this challenge and ensure that reagents are accessible and reagents are also affordable to the public health institutions in the continent. Most importantly, technical support when it comes to installation and technical support for maintenance, training. That is also a really critical part of our partnership and our discussion with the private sectors.
Excellent. And we actually look forward to seeing how that matures over time. I don't see any questions from the floor. I think at this stage, Sophonis, I want to say thank you very much for your presentation. And then it leaves me then with a pleasure to hand over to Duncan McCannell, who is the co-PI on the Phage project and who's based at the US CDC, and he will do the closing remarks. Thanks very much, Duncan.
Thanks so much, Alan. This has been a fantastic first open meeting, and I'd like to thank everyone for joining us today for the first open meeting of the Public Health Alliance for Genomic Epidemiology. This week marks 2 years since a group of public health bioinformaticians, epidemiologists, and laboratory scientists first gathered in Seattle from across the world. And at the time, there was really no specific agenda. There was just a single, perhaps idealistic question, and that was, how do you begin to improve the openness, interoperability, sustainability, reproducibility, and access across global public health bioinformatics? I guess that's a compound question. But this really began the dialogue, and Phage was the first result of these discussions. Pathogen genomics and genomic epidemiology are transformational for global public health, and bioinformatics skills and infrastructure and resources are rapidly becoming one of the most important bottlenecks. There are important gaps in software and pipeline development, the establishment of standards, and the ways that we develop and integrate tools into platforms. And the past year has really demonstrated the importance of improving, expanding, and investing in the global public health laboratory, bioinformatics, molecular epidemiology, and the broader data workforce around it. By focusing on the tools, the standards, and the expertise that we need to generate, analyze, and use the data, we think that we can improve the interoperability, the sustainability, the access, the sharing of benefits, and the impact of these technologies at a global level. Because importantly, what you come to realize is that so many of the pressing challenges that we face are incredibly universal. Virtually all countries at some level face the exact same problems around implementation and sustainability of pathogen genomics and bioinformatics needed to make it work. And these shared challenges, while they are daunting, they do present an important window of opportunity. I'd like to thank Alan and Alicia and Jamie and the entire Phage Secretariat for organizing an exceptional first meeting. I'd like to thank the presenters today who showcased so much of the excellent work already going on in the work groups and some of the products that have come out of those discussions and efforts. And I'd like to thank all of the work group leads and the work group participants for their hard work and their enthusiasm over the past year. And it has been a difficult and but at the same time, important year. And I'd like to close this meeting, if I could, with a final call for action. We would love for you to come join us. If you want to join a working group, there are instructions on the website. There are many areas where we could use different ideas and expertise and perspective. If you want to just follow along with some of the developments and the projects, or incorporate some of the outputs of the Phage groups into the work that you do, you can follow Phage on Twitter or GitHub or a variety of different other social channels. But overall, thank you for joining us today, and we hope to hear from you all soon.
Thanks a lot, Duncan, and we will definitely be in touch with all of you. Enjoy and keep safe.